Okay, good morning, everyone, or good afternoon. Welcome to Ag Science Café for December. It's already end of year. Amazing. So I'm Cherry Kubota. If you are new to this space, I'm the moderator and then one of the organizers of this cafe series. We have been doing I probably 6, 7 years now as an effort of the multi-state project, now funded by USDA, few groups, ADVANCEA, OptimIA, and CEA HERB, basically sharing science-based information to move the CEA industry forward. So that's basically the idea. So we are doing monthly, Tuesdays, 11 a.m. is our standard time, Eastern time, and then selected Tuesday usually. So if you are new, just expect that once a month, one of the Tuesdays of the month is going to be indoors Ag Science Cafe. So we are accepting suggestions of speakers. I show you the upcoming schedule, but we like to have industry speakers as well as academic speakers. Academic speakers can be a group presentation like today, project funded or funded project based speaking team would be great. Or graduate students or postdoc wanted to share the outcomes together with their supervisor. That would be also great. Industry like to hear the status, challenges, may not be so willing to share the challenges, but if it is everyone's problem, that's easier to share, and then we can work on that. So that's some of the opportunities I wanted to emphasize. So upcoming one, so the today's one I'm going to explain in a minute. And then January, we have Melanie Yelton, who is a consultant, and then forming Growers Association for CEA. So that's a great effort, and wanted to know more about that effort and contents. So that's January 20th, Tuesday, 11 a.m. And February, we're going to have Ian Justice, who is right now, I think he's working in the cannabis industry, but he has long history of berry production, small fruit production in commercial settings under controlled environment. And he's now doing consultant, separate sort of work to serve for other CEA growers. So he's going to share the tips and important topics regarding raspberry and blueberry. Raspberry and blackberry production under controlled environment. And in March Cafe, we are actually in communication with the grower or the founder of a company who does indoor production vertical farm. So I haven't confirmed, so I can't disclose who it is, but hopefully this is happening in March or soon after. Okay, so today we're gonna have three talented scientists talking about a project funded by USDA NIFA, Urban Indoor Emerging Agriculture Program. And then the project title is Tailoring Hydroponic Factors to Control the Environment Production of Emerging Food Crops. So it's very interesting. Dr. Qingwu or William Meng, University of Delaware, or Dr. Yujin Park, Arizona State University, and Dr. Joshua Craver, Colorado State University. They are going to give three presentations, and then I'm not gonna read the titles. You get the introduction by them. So with that, it's so exciting to have group presentation like that. And then I really appreciate for three of you. And then I'm going to stop sharing and then starting with William for his introduction of the project and then his project talk. Interesting biostimulant. All right.
All right. Thank you so much, Sherry, for this great opportunity. And hello, everyone. Welcome to the Indoor Ag Science Cafe. Like Sherry said, my name is Qingwu William Meng. I'm an assistant professor of controlled environment horticulture at the University of Delaware. And today, like Sherry said, I'm going to talk about our USDA NIFA funded project from the Urban Indoor and Emerging Agriculture Program, or UIE. And this is a project that we have been working on since 2023 and it's still ongoing. It's a combination of research and education. So the title is Tailoring Hydroponic Factors to Control Environment Production of Emerging Food Crops. So we're trying to look at not only the most popular crops like lettuce, but other emerging leafy greens such as spinach and arugula, as well as small fruiting crops such as strawberries and peppers. I am the lead project director, and working with me are also our co-project directors, Dr. Yujin Park from Arizona State and Dr. Joshua Craver from Colorado State University. These are our research objectives. I can put them into 3 buckets. The first one is to look at the physical properties of hydroponic nutrient solution management. And Yujin and I have been working on the dissolved oxygen and temperature control. The second bucket is chemical properties of hydroponics. And I work with Yujin on evaluating novel chemical-based biostimulants in hydroponic nutrient solution application, as well as fine-tuning fertilization to improve nutrient use efficiency. And lastly, we also look at physiological disorders in crops, such as tip burn in lettuce and intumescence in peppers. And this is an area that I and Josh work on at. The 3 of us also work together on achieving these educational objectives, and we saw a lack of open source, adaptable teaching materials for higher education of hydroponics and controlled environment agriculture in general, beyond the traditional textbooks that we had. So we wanted to have an open source, open access model where we can upload and share the teaching modules, especially geared towards experiential and hands-on learning experiences for undergraduate level teaching. So we have been going through the developmental phase, creating the standard operating procedures, collecting videos and images to illustrate the hands-on activities for learning about hydroponic system design, plant propagation, nutrient solution management, light management, and selecting and growing and managing different crop types. And the next phase of this area will be to implement our teaching modules in our own classes at the 3 universities and develop assessment models to see how that improves student learning outcomes. So now I want to focus on my research at the Delaware Indoor Ag Lab at the University of Delaware. I have been here since May 2020, so this is our 6th year, and we'll keep going. And we have these facilities showing these pictures, including the indoor rooms, growth chambers, and greenhouses to work on a variety of different projects on specialty crops. And one particular aspect that I emphasize on in this particular research collaboration is to address lettuce tip burn, which is a physiological disorder caused by localized calcium deficiency. And that happens at the shoot tip of lettuce, as shown in this picture, when we have an imbalance between calcium demand exceeding calcium supply, because calcium is an immobile element. And oftentimes, we have enough calcium ions in the nutrient solution, but plants just aren't simply able to take up and mobilize calcium to the tip of the shoot where calcium is needed the most. And that is typically limited by environmental conditions, such as the environmental conditions that can make transpiration rates low, such as high humidity and low airflow. So there have been many strategies devised to improve transpiration and thus mass flow driven calcium uptake and mobilization in order to mitigate tip burn. But they all have their pros and cons. So recently, in the past several years, I've had this wonderful collaboration with my collaborators, including Dr. Susan Sun, who I see is in the audience as well. And this is a chemical company from the UK, but they also have large presence in the US. And we've been testing different novel and sustainable chemical-based, potentially biostimulants in hydroponic nutrient solutions to see if we're able to improve nutrient use efficiency and mitigate disorders such as tip burn. So this particular product is codenamed CCUS 2105. It was developed with the calcium mobility technology to facilitate the movement and mobilization of calcium within the plant. Originally, it was developed as a foliar spray to alleviate calcium deficiency in field crops, but there was no test of this product in hydroponic nutrient solution. So we wanted to do this directly by applying a small amount of this chemical fertilizer in the nutrient solution for deep water culture production. And we did a previous trial to identify the optimal concentration of this biostimulant. As we can see for these two lettuce cultivars, Rex on the top and Dragoon at the bottom, increasing the biostimulant concentration from 0 to 1 milliliter per liter mitigated tip burn. And at the same time, we also saw a difference in plant growth and plant size. We cannot have too much of this biostimulant, otherwise we can have stunted plant growth, as seen under the highest concentration here. The best optimum concentration seemed to be 0.5 milliliters per liter in this case, where we saw a great reduction in tip burn severity and incidence, as well as yield being maintained compared to the control. So to follow up on that, we wondered if this biostimulant would compare to the traditionally effective vertical airflow fans, which act to disperse the humid air around the center of the plant and improve airflow and reduce the boundary layer, and in order to increase the calcium mobility within the plant. So we did this experiment with my former lab graduate student intern, Moin Musavi Nashat. He's now finishing his PhD at NC State with Dr. Ricardo Hernandez. This experiment was conducted at relatively high temperature, humidity, and daily light integral, as shown here. And we had the split block design with 2 blocks. We grew some plants under no vertical airflow fans and others under vertical airflow fans, providing the vertical airspeed of 1 meter per second. And within each fan category, we had biostimulant concentrations of 0, meaning no biostimulant acting as a control, 0.25, and 0.5 milliliter per liter, based on previous research. And these are the plant responses. So 14 days after transplant, we saw no tip burn in any of the treatments, and plants looked comparable in size as well. 21 days after transplant, we saw tip burn showing under the control, where plants did not receive either the biostimulant or vertical airflow. And if we increased the concentration of the biostimulant, we saw great reduction in tip burn. At the same time, regardless of the biostimulant, the vertical flow fans themselves were sufficient at eliminating tip burn. And finally, at the last harvest, at 28 days after transplant, the same trend continued. We saw severe tip burn under the control. Either the biostimulant or the vertical airflow fans were sufficient at mitigating tip burn. These were the data, and we can see here for tip burn mitigation, we're quantifying that with the tip burn rating on the left and the percentage of leaves with tip burn on the right. So in this case, we saw reduction of tip burn by 94 to 96% at 21 days after transplant, and 71 to 75% at 28 days after transplant. So at the final harvest, we achieved comparable tip burn suppression compared to vertical airflow fans if we were to use the biostimulant at the 0.5 milliliter per liter concentration. And that was all done with no effects on the final yield, as we can see in this slide here. On the left, we have shoot fresh mass. On the right, we have shoot dry mass. at 21 or 28 days after transplant, with no vertical airflow, the biostimulant did not affect the biomass accumulation, which was good news. We also evaluated the inner tissue nutrient concentration for the lettuce to see how exactly the nutrients were mobilized, especially calcium. So in this case, on the axis, we have the inner tissue calcium concentration as the percentage of dry mass. And on the y-axis, we either have the tip burn rating or the percent of leaves with tip burn. So the higher these numbers are, the more severe tip burn was. And we saw a negative correlation between the inner tissue calcium concentration and tip burn severity. So as we increase the biostimulant concentration, we saw a reduction in tip burn, and that's correlated with increased inner tissue calcium concentration. So this supports the hypothesis that tip burn was caused by localized calcium deficiency, and this biostimulant was able to mobilize calcium through the symplastic pathway to aid the typical apoplastic pathway for better calcium mobilization. If we have this biostimulant with no vertical airflow, we're able to have comparable and sufficiently high calcium concentration in the inner tissue compared to the plants grown under the vertical airflow fence, which again, by themselves are sufficient to get enough calcium to the center of the plant and minimize tip burn. But again, fans can be bulky to install. They also consume electricity. So when that's not practical, the biostimulant can be a really great tool to help with tip burn mitigation. And nothing beats a picture. It says 1,000 words. So this is a picture taken of all the plants side by side at 28 days after transplant. And we can clearly see how bad tip burn got under the control with no vertical airflow or the biostimulant. And if we use this biostimulant with no vertical airflow, we could achieve comparable tip burn mitigation and no yield penalty compared to the vertical airflow fence treated plants. And we have published this research this year, earlier in the trade magazine Inside Grower, as a grower-oriented article to summarize our biostimulant research in collaboration with Croda. And just a couple weeks ago, Meng and I have published this research in Frontiers in Plant Science. So if you're interested in reading more about the details for this research, you can scan the QR codes here. And lastly, I want to follow up on another study we did last year to look at how this biostimulant could potentially be applied to other leafy greens, such as arugula, kale, and pak choi. And we saw that not all crops responded the same. So for arugula, kale, and pak choi, the biostimulant actually had a negative effect on their shoot fresh mass at 21 days after transplant. But plants recovered a week later, and there was no difference with or without the biostimulant in terms of shoot fresh mass for those crops. And encouragingly, we saw that the biostimulant also improved the shoot fresh mass of lettuce at both harvest dates. And these were the data to support those picture observations. And at 21 or 28 days after transplant, we saw a yield increase in lettuce by 23 to 29%. And all the other crops tested here, arugula, kale, and pak choi, they happen to be in the same Brassicaceae family. They're all cruciferous vegetables. So it's interesting that they all responded similarly to this biostimulant. So maybe future research can test lower doses and see how they will respond. And now this is a segue for Yujin's presentation. We have collaborated on this research and recently published this paper on improving the baby spinach germination and subsequent growth in deep water culture hydroponics by manipulating the substrate moisture as well as the active aeration and nitrogen concentration during the production phase. So if you're interested, you can scan this QR code to learn about this research, but Yujin will talk more about active aeration or dissolved oxygen on her side. And finally, I would like to acknowledge the funding support from USDA NIFA and UD, as well as the members and greenhouse staff from UD and my lab for assistance, And finally, this could have not been done without the support from our industry collaborators, including CRODA. So thank you, Susan, especially, as well as Groden, GR Peters, and Vault Grow. And if you're curious about any of the research I talked about or want to follow up, please feel free to email me here. Thank you.
Thank you, Qingwu, for your presentation. Hello, everyone. My name is Yujin Park. I'm an assistant professor at ASU. And as part of USDA UI project, my research team at ASU has been investigating how dissolved oxygen management affect the crop growth in hydroponics. And I will share the findings from our study. So dissolved oxygen refers to the amount of oxygen present in water, and in hydroponics, it's important to maintain a proper level of DO for root growth, nutrient uptake, and disease prevention. And it's recommended to keep the DO level close to the water saturation point, which typically ranges between 7 to 9 ppm, depending on the nutrient solution temperature. And as you can see in this diagram for deep water culture and nutrient film technique, the air pump connected to the air source are commonly used to aerate the nutrient solution and thus to provide the dissolved oxygen to the nutrient solution and the plant roots. However, when we manage nutrient solution, we often prioritize to manage other hydroponic factors such as pH level, EC level, and sometimes we overlook the importance of DO management, and then we don't regularly monitor DO. So in the hydroponic system, the nutrient solutions are often insufficiently aerated, and especially, as you can see in this photo, the multi-layer hydroponic system. In this multi-layered hydroponic system, the DO concentration gradient can easily form as the aerated nutrient solution is delivered first to the top growing tray, and then consequently, the lower growing tray receives the nutrient solution with the reduced dissolved oxygen concentration since plant roots uptake oxygen. And that's actually what happened in my lab when we grew the strawberries. So at first, we didn't monitor DO level and we didn't pay attention to DO. And after we observed some growth differences in strawberry, later we realized that it was probably related to the lower DO level in the lower growing tray. And since then, we have been interested in learning more about how DO management affects plant growth in hydroponic systems. So in this talk, I will present the findings from two of our experiments, one with strawberries and another with leafy greens with spinach and arugula. So the first study, we focused on strawberry, and here we examined how supplementing DO affect the plant growth and fruit production in strawberry. So for this study, we grew strawberry albion and eversweet cultivar inside the indoor vertical farm with the following condition. And we use a deep water culture system that have a growing tray on the top and a reservoir in the bottom. And we tested three conditions. So for control, we provided aeration in the reservoir using an air stone connected to the air pump. And to create the DO supplementation condition, we added the air stone in the growing tray. And then we connected the air stone either with air pump or oxygen concentrator. And the major differences of using air pump or oxygen concentrator was the DO concentration delivered, because both air pump oxygen concentrator, they take in ambient air, but air pump, they pump out ambient air as it is, which is 21% of oxygen concentration, while oxygen concentrator, they concentrate oxygen inside the device and then pump out concentrated oxygen with high concentration, typically 90 to 95%. So by using this setup, we were able to create three different DO concentrations. So when we monitor the DO level throughout the experimental period, the average DO was 6 ppm for control and 7.5 ppm when we supplemented DO using air pump, and 11.3 ppm when we used oxygen concentrator to supplement DO. And in terms of DO saturation percentage, this number corresponded to 67%, 83%, and 126% for respective conditions. And the first observation we made about the effects of DO supplementation was on root initiation. So after transplanting strawberry bareroot plants in the deep water culture system, new roots began developing within a few days. And we monitored the timing of root initiation. And it was quite straightforward to identify the new roots because new roots looks white compared to old roots looks brown. And our results show that increasing DO concentration from 6 to 11.3 ppm decreased the days to root initiation by 2 days in both cultivars. And after root initiation during the vegetative growth stage, we saw the effects of DO supplementation on shoot growth. So this photo shows the albion cultivar on the top and eversweet cultivar on the bottom, and the plants were grown for 6 weeks under DO treatments. And then the photo clearly shows that increasing DO concentration enhanced the shoot growth. And when we evaluated the leaf number, leaf area, shoot fresh mass, and dry mass, we observed all these 4 growth parameters linearly increase with increasing DO level. And between these two cultivar, albium responded more sensitively to the DO supplementation. So the magnitude of increase in plant growth parameter was greater in albion than eversweet. And given the effects of DO supplementation on shoot growth, we also expected the similar effects on root growth. But we observed root fresh mass, dry mass, and root length was not affected by DO supplementation. And the only noticeable response was root fraction, when we calculated as root fresh mass divided by the total fresh mass of root and shoot. So root fraction linearly decreased with increasing DO level, indicating that DO supplementation did not affect the absolute root growth, but shifted the biomass of allocation more toward the shoot over the roots. Additionally, we hypothesized that increasing DO level would enhance the root nutrient uptake, but when we performed the plant tissue analysis, the concentration of all macro and micronutrients in the plant tissue level was not affected by DO level. And after vegetative growth, we also evaluated the flowering time, and increasing DO concentration accelerate the flowering in albion, so the days to flowering decreased by 5 days, although this trend was not observed in eversweet. And another focus here was to see the effects of DO supplementation on fruit production. But when we evaluated the parameter related to the fruit yield and fruit quality, including days to fruit harvest, total fruit numbers, total fruit yield, and individual fruit fresh mass, individual fruit diameter, and roots value, we found there was no effect of DO supplementation on all of these parameters. So in summary, when we examine the effects of DO supplementation on plant growth and fruit production in strawberries, we observed the DO supplementation promoted the root initiation and shoot growth, but had limited effects on fruit production. So our results suggest that the optimum DO application for indoor strawberry production may be best to focus on promoting vegetative growth. Then next, I would like to share our experiment with leafy greens, where we focus on spinach and arugula. And here, we wanted to better understand how the way we deliver the oxygen affected the DO concentration in hydroponic system setup and then plant growth. So here we grew arugula astro and spinach ouch, similar in the indoor vertical farm conditions. And we use the same deep water culture system setup that has one growing tray on the top and then reservoir on the bottom. And in terms of oxygen delivery, we tested the aeration method and location. So we compared 2 aeration methods, either using oxygen concentrator or air pump. And then we provided the aeration either in the reservoir, growing tray, or both locations. So this diagram shows our treatment design. So we totally had 6 treatments. And in this study control, we didn't provide any aeration either in the growing tray or reservoir. And 2-3 months, we used oxygen concentrator to provide aeration. And then aeration was provided in the reservoir or growing tray. So here the green area indicates the aeration location. And 3 months, we use air pump to provide aeration, and then the aeration was given in the reservoir, growing tray, or in both locations. And the number in the growing tray indicate the monitored DO level under each aeration condition. And as expected, the control had the lowest DO value. So it was 5.6 milligram per liter throughout the experimental period. And when we use oxygen concentrator, DO level increased up to 9.5 milligram per liter when aeration was given in the reservoir. And it was 19.7 milligram per liter when aeration was given to the growing tray. And when we use air pump, when the aeration was given in the reservoir, the DO level was similar to control. So the average DO level was 6 milligram per liter. But when air pump provided aeration in the growing tray or in both locations, we observed the DO level increased up to 7.3 milligram per liter. So overall, using oxygen concentrator and providing aeration in the growing tray compared to reservoir was more effective at increasing DO level. And here, I have a photo for arugula and spinach, which grown for 4 weeks under 6 aeration treatments. And then we can see both arugula spinach grow larger when aeration was given using air pump in the growing tray alone or with reservoir. And arugula also grew bigger when the aeration was given in the growing tray using oxygen concentrate. And I have shoot fresh mass data here, and then we can see the same trend in shoot fresh mass quantitatively. So when aeration was given in the growing tray or in the both growing tray or reservoir, it increased the shoot fresh mass in arugula spinach more than 100%. And also in arugula, when aeration was given using oxygen concentrator in the growing tray, it increased the shoot fresh mass by 223%. So in this experiment, when we evaluated the impacts of aeration method and location on the DO concentration in hydroponic nutrient solution and shoot fresh mass, we found that oxygen concentrator generate higher DO level than air pump, and direct growing tray aeration produce higher root zone DO than reservoir aeration. But in terms of the plant growth, air pump aeration in the growing tray most effectively improved shoot freshness. Then I would like to acknowledge the contribution of my student, Joasan, Keith, and Nicholas, who played an essential role in conducting experiment, and we thank USDA for their great support.
Excellent. Thank you so much. And so my name is Joshua Craver, Associate Professor at Colorado State University. I do want to go ahead and just apologize in advance. I'm getting over a nasty cold over the last few days, and my voice is just now starting to come back. So if I break up a little bit and the audio quality's bad, again, apologies. But today, excited and thank you again for inviting us to be here. But I'm looking forward to talking about some of the research we've been doing regarding intumescence development on pepper, looking specifically at some hot pepper varieties, as well as a sweet pepper variety. So understanding and mitigation of that disorder. Before I get too much into the details there in terms of the research we've been doing, I just want to first acknowledge the facilities where we've been conducting this work. And so first of all, being the CSU Hort Center, this is located in Fort Collins. I am the director of this facility, but I work very closely with our manager, Mike Hazlett, whose photos over here on the left. I've been doing lots of research in this space, 21,000 square foot greenhouse. So really grateful for this facility. And then also the CSU Spur campus that has also been supporting a lot of the research, as well as particularly the education components from CSU's perspective. And so we have a controlled environments program there with a rooftop greenhouse and a lab and growth chambers. And so a quick shout out to Dr. Jay Colley, who manages those facilities for us. And so with that, I want to go ahead and jump into intumescence development. And so I know some of us may be familiar with it, may or may not be familiar with this disorder, but it's a physiological disorder that is not new by any means. This is actually a disorder that was first identified late in the 1800s. It's just become a little bit more center stage over the previous years. Because of the push in terms of controlled environment production. As we're producing more crops in controlled environments, we're seeing the incidence of intumescence just popping up a little bit more. And so what intumescence is it is characterized by abnormal growth of epidermal as well as palisade and spongy parenchyma cells. And so what you get is these cells that ultimately undergo hypertrophy or cellular expansion, as well as hyperplasia, cell division. And this occurs rapidly until you get these abnormal growths or lesions that develop on the surface of plant tissues. And so a great example you can see here at the bottom is for ornamental sweet potato. And so you can see those large translucent, in this case, growths that are developing along those leaf veins. As we look at a cross section specifically of these leaves, though, we can actually see where that cellular growth is occurring. And so if we look at the photo over here on the left of this cross section, you can see the upper epidermis, the palisade parenchyma, spongy parenchyma, and the lower epidermis, again, of that sweet potato leaf. And again, normal looking leaf, no lesion to be seen. And then if we look at the photo on the right, again, you can see the presence of both that hypertrophy of cells as well as the hyperplasia as we get this elongation and expansion that leads to lesion development. Now, there are many, excuse me, many different plant species that are ultimately impacted by this disorder. A lot of them are ornamental. So we do see a lot of ornamental species that are going to be susceptible, which causes a lot of issues with their aesthetic value. But we also see vegetable crops in controlled environments, specifically the solanaceous family. Solanaceous crops are relatively susceptible. You can see pepper as well as tomato here in my photos on the bottom. And so if we start with pepper, you can see a photo that we took at the Hort Center over here of one of our pepper plants, and you see those leaves starting to curl. And as we flip those leaves over, you can see, again, the development of those lesions, that intumescence on that abaxial or underside surface of that leaf. Now, if we look over at the photos on the right, you can see tomato. And we see very similar patterns in terms of the disorder. You can see some early stages of development in tomato, where again, we get those extrusions of those lesions developing on the underside, again, abaxial surface of the leaf. And then as development continues, you'll actually see that those lesions begin to collapse and actually lead to some large senescent areas on the surface of the leaf. Now, one of the big questions that we then have to ask is what is actually causing this disorder to occur? And this has been the product of the question that's asked for a lot of the research that's been conducted over the decades, trying to really understand what's leading to this abnormal cellular growth. And so there's been many different factors that have been associated, ranging from water status in the leaf, leading to excess cellular pressure, to other factors that may be contributing to expediting this disorder, such as changes in relative humidity, specifically high relative humidities leading to a reduction in transpiration. Often high temperatures have been associated in terms of excess water uptake, as well as lighting, both light quality as well as light intensity. Now, one thing that's really interesting about intumescence, though, that helps us as we try to elucidate what the causative factors are that are driving the disorder, is when we go out into the field and look in the outdoor environments, we don't typically see this disorder occurring. It's only when we move those crops into a controlled environment that we start to see the development of intumescence. And so you start to think about what are those environmental factors that differ between this indoor controlled environment and the crops we're producing outdoors. And one of the low-hanging fruits of what may be a causative factor early on was UV radiation. Specifically, if you think about greenhouses, we have UV blocking additives that we add to our glazing materials. And so as a result, we have the lack of UV in these controlled environments. And so it was thought that possibly the lack of UV in these spaces was leading to the development of intumescence. So there was a lot of research done looking at how we can supplement in and try to remedy or supplement UV to try to remedy the development of this disorder. But again, a lot of research that's been done on lighting as well as other environmental factors. And I would say at this point, it's still a little bit elusive in terms of what is causing the disorder to occur and what specifically is happening at that cellular level that's driving this hypertrophy and hyperplasia of those cells. And so This then led to the research that we wanted to conduct as part of this USDA project. And so specifically looking at pepper, we were really curious in better understanding how we could leverage lighting treatments to try to prevent and try to mitigate the development of this disorder. And so specifically, we were interested in really low intensity lighting application treatments, so end of day or pre-dawn low intensity light. But before we wanted to move forward with looking at those lighting treatments, we wanted to really understand when cellular growth in peppers was at its greatest, when we saw the most, for example, leaf expansion occurring in those upper pepper leaves. And so our first objective was really trying to understand the timing of leaf expansion so that we could understand how that coincides with the development of intumescence, so that we could then lead on to objective 2, where we are applying those lighting treatments at appropriate times to hopefully suppress the disorder, but also to look at how that light quality and that light intensity we're providing may be impacting pepper morphology as well as pepper growth. Now, what you'll notice is that then we move into our objective 3, which is completely different from the other two, where we begin looking at water stress. And so I'll get to that in just a minute, so we can talk a little bit more about why water stress really became a high priority item in terms of our research moving forward. So we'll first start by just really briefly looking at this first objective. So over here on the excuse me, over here on the right, we have Courtney Dunbar. She's my master's student that was working on this project over the past couple of years, just graduated this past fall semester. But what she was doing for this first evaluation of pepper leaf area expansion was looking at 3 different cultivars. We have California Wonder, that was a sweet pepper cultivar, as well as 2 hot pepper cultivars, Potapino and Spicy Jane, that we got as recommendations from Pan American Seed. So we evaluated lots of cultivars, found that these 3 were relatively susceptible to the disorder, and moved forward with some experiments to try to characterize that leaf area expansion. And so what we did in this instance is Courtney went through and actually tagged individual leaves on these pepper plants that were undergoing active expansion and evaluated that expansion and many other characteristics of those leaves over the course of a 1-week period. And so she would come in twice a day at 7 a.m. and 7 p.m. and take images of those plants that we could then use to derive a lot of measurements of those leaves, including leaf area, which you'll see on the next slide. And so here we have 2 figures for California Wonder and Spicy Jane. Due to time and space, I'm just going to talk about those 2 cultivars, but Potapino followed very similar trends. But what we can see is in these figures, we have 6 days represented here, as well as leaf area difference. And so leaf area difference was our way of looking at, in this case, what differences occurred in leaf area during the day compared to the night. So you'll see over here that our orange dots are during the day from 7 a.m. to 7 p.m. And the blue dots are going to be during the night from 7 p.m. to 7 a.m. the following morning. And what we found was that most of that leaf area expansion was occurring for all 3 cultivars during the evening. We actually found that over the course of that 7-day experiment, that it was about 94% greater leaf area expansion during the night compared to the day for California Wonder, and about 46% greater expansion during the night compared to the day for Spicy Jane. And so again, we were hoping that this would be able to help us better predict when we may want to be applying our lighting treatments, and also evaluating and looking for that development of intumescence for targeting those lighting treatments. However, after conducting some of the lighting treatments, which I'm going to pass over today due to time, we saw some changes in terms of pepper morphology. But weren't really finding a good degree of success in terms of mitigating the disorder. And over the course of a couple of additional experiments, what we found was that water stress seemed to really be playing a role. It seemed to be somewhat of a trigger for these pepper cultivars in terms of whether or not they were going to develop intumescence. And so, at least for the cultivars that we were growing in our greenhouse, the environmental conditions that we provided them, water was a very predictable trigger. And I'll show you that in this next experiment, which was a very straightforward experiment that we conducted as we were starting to evaluate whether water may be playing a role. And so we again had the same 3 cultivars. And in this case, Courtney set up just 2 very simple water treatments. We had a water stress treatment, is what we're calling this, where we allowed those plants to dry down to 40% of their container capacity. This was a one-time water stress event where we started to see some early signs of that wilting, flagging from those plants. And we followed this up by what we're calling a watering event, where we rewatered those plants back up to their full container capacity. The control plants, we did not allow to be subjected to any water stress. They were watered consistently at 70% of their container capacity to make sure that they never experienced any of the drought symptoms or water stress. And so what Courtney did for this study was came in again 2 times a day. She would come in at 9 o'clock in the morning and 5 o'clock in the afternoon to weigh plants. And we were using those weights to determine when we needed to apply our water treatments and rewater our plants. And additionally, at those time points, Courtney would come in and evaluate all the leaves of the plants in both treatments across all 3 cultivars to look for any signs of intumescence development. And so that's what you can see in this figure here on the bottom is we have intumescence occurrence on the y-axis. And so that's the percentage of plants showing symptoms of intumescence. There was an N of 10 specifically for this study for each cultivar and each treatment. And then we have observation date. So you can see that 4 days prior, as well as 6 days post that watering event for those water-stressed plants, we were evaluating those plants for signs of intumescence development. So 4 days prior and 4 days post. So if we then look at some of the data, it's actually really telling, at least for 2 of the cultivars. And so if we now look at specifically the control or this blue line where the plants were not allowed to be subjected to water stress, we see that over the course of this 10-day observation period, no signs of intumescence were observed on any of the cultivars. Where we start to see some really interesting results is specifically with the Spicy Jane. And unfortunately, I don't have the Potapino on this slide as well, but Potapino and Spicy Jane were almost mirror images of one another in terms of their response to this water stress treatment and this increase in intumescence. And so what we see is that for Spicy Jane, after that water event, after those plants were rewatered after experiencing that water stress, within 2 days, 100% of those plants were then showing symptoms of intumescence. And so we had intumescence developing on many of those upper leaves that were undergoing rapid expansion. And again, 100% of plants for Spicy Jane and Potapino were showing symptoms within 2 days after that water stress event. For California Wonder, we didn't see nearly as many plants showing symptoms. It was actually only about 20% or so of the plants that were showing symptoms. And it was about 3 days after that water event. And so we think there may be a couple of factors at play here, one of which being California Wonder was a little bit more resistant to that water stress than the other 2 cultivars in terms of showing symptoms to that water stress event and flagging. And so what we're going to be doing moving forward with some future experiments is upping some of our water treatments, doing a little bit more intensive job of tracking water status in the plant using soil moisture probes. And so you can see I have a student that's currently working with a lab here on campus to build some of those in-house because we have a lab that specializes in a lot of this. But I do want to note just really quickly that environmental conditions were relatively stable over the course of this experiment, which is really important for intumescence development because there's so many environmental factors that could be implicated in the development of this disorder. We want to make sure that that environment remains relatively consistent. The only factor that hopefully is changing is that water stress. And so not just this experiment, but in experiments since we've run this, we found that this is a pretty predictable trigger where if plants are not subjected to water stress, we don't get intumescence, but upon subjection to that water stress, we're pretty predictably seeing symptoms of the disorder. So we have a handful of experiments planned moving forward that we're really excited about, as well as a lot of work like William mentioned regarding the assessment of some of these hydroponic modules that we're really excited about the development of. So we look forward to sharing more of those results with you all in the coming months and year. But with that, just a couple of quick acknowledgments. Do really appreciate Courtney as well as Matt, my new master's student who's been working on this project now that Courtney's graduated, as well as in particular Pan American for working pretty closely with us in terms of getting a seed and some of these cultivar selections. And so with that, I think that wraps us up for the presentations.
Wow. Thank you so much. Such a great combinations of different topics and different factors. I really appreciate William and Eugene and Josh for the great presentations and sharing the outcomes.